Hi again, and welcome to Chapter 4, Part 2. We are going to pick up where we left off, talking about enzymes and chemical reactions. How do we control reactions with enzymes, and how do we control the enzymes, so controlling the controllers? As you may recall, what enzymes do is they lower activation energy. That makes a reaction more spontaneous by making it more sensitive to the energy in the situation, in the environment that's already there. You don't have to add any additional energy to what's already there in the environment. Another way of saying that is making the reaction more temperature sensitive, because temperature is in fact a form of energy. It's a kinetic energy of the vibration inside the molecules and atoms of the reactant that you're dealing with. So making it more spontaneous by making it more sensitive to the energy that's already there. So how do we control the controllers? How do we control enzymes? And I've got a laundry list here, and we're going to go through each of the items on this list, give you some examples. One is the presence or absence or the amount of the enzyme. We call this generally up and down regulation. There are some ways of saying up and down regulation that talk about the activity of the enzyme, but for our purposes for this class, we're going to say abundance of the enzyme. We can add or remove modulators that will change the reaction rates. That may be something like a product in the metabolic pathway. We may modify the protein some way with what we call cofactors or other types of modification, and then cofactors themselves. These are molecules that influence the activity of a given enzyme, usually in a positive way. Using different enzymes to catalyze reversible reactions. This is a little bit of a tougher concept. I'll get there or I'll tell you about it when I get there. But our model system is going to be kinase and phosphatase. So if you want to look up those terms, that's a great place to start if you're having trouble understanding what we mean by using different enzymes to catalyze reversible reactions. What we mean to say is we're catalyzing only one direction of a reaction with a kinase. And that same reaction, we're only catalyzing one direction of the reaction with a phosphatase. Compartmentalizing enzymes within organelles, using the cytoskeleton or using organelles in their membranes or vesicles, a spatial regulation of enzymes, either within compartments or just out in the space inside a cell, keeping those enzymes away from where the reaction might occur. This last one is a little bit like cofactors. It's a little bit like point number 2. A lot of enzymes are actually quite sensitive to the ratio, not the overall abundance, but the ratio of ATP to ADP. And that implies is that these molecules can and do bind to the proteins just like cofactors do. So it's a little bit of a corollary, but a very, very important one, because cells can and do alter their activity based on this ATP to ADP ratio. And look no further than caffeine. Caffeine actually works alongside this phenomenon to fool the cell into thinking it needs to make more ATP, and thus ramping up activity. And let's start with point number 1 of our list. I'm going to be going back to that list. You might see it in your PDF, your PowerPoint handout, that I'm going to be referring back to that previous slide. Upregulation and downregulation. For our purposes, upregulation means there is more abundance of a given protein or an enzyme in this case. We'll say those are synonymous in this situation. And this is an example. This is called a Western blot. What we do is we have an antibody that's relatively specific for a given protein or enzyme. And you can see that in column A we have a certain amount here. And in B, it looks like we have more of it because it's darker, it's more intense, even a little bit bigger. That infers or implies more abundance of that biomolecule. If it's an enzyme, that means that the reaction is probably going to proceed more quickly because you have more catalyst there. How do we get from here? Well, there's lots of ways, and it all has to do with making proteins, because most of these enzymes are proteins. Maybe we transcribe more of the gene from the DNA, so we make more messenger RNA. Maybe we translate more of it. And there are examples of genes that, like in the case of inflammatory genes, where you have lots and lots of the messenger RNA, but you don't translate it until you need it. And then the translation kicks up right away and you make a lot of inflammatory proteins. So how fast do you translate? So on the creation side of the protein, we can either make more messenger RNA or we can translate more of that messenger RNA. Or we can decrease the rate of degradation of that protein. Proteins generally live a relatively finite life, especially soluble proteins, proteins that are enzymes and involved in chemical reactions. They wear out or they're just degraded over a period of time. If we can slow that degradation down, that means more protein sticks around relative to how fast we're creating it. Then the down regulation side. It's just the opposite. So we have a protein here that we can see, and you can see it's going away. There's less of it in, less obvious band there. Less transcription, we shut the gene down. Less translation, or we increase the rate of degradation. So it's like a teeter-totter between creation and destruction. And so we have a figure here from your book. This is page 113, and I'm not going to test you on all the steps because you had this in 165. So you should have some awareness of it, and the idea of turning on and transcribing the gene, so how much we transcribe. Now, mRNA processing is sort of on that same side. If you process the mRNA, you're going to end up with more mRNA around for longer. It doesn't go away as quickly, which means more mRNA. On the other hand, how fast we translate it. And then at the end here, what happens to the protein after we translate it? Is it folded? Do we cross-link it and do other things? And we have a whole series of things we're going to talk about here. There's a lot to discuss. This figure is on page 113. The key is just to be able to understand what's going on through these steps and understand why they would be either on the creation side or in the case of cleavage on the destruction side. And so we've talked about manipulating enzyme concentrations, up and down regulation. Now let's talk about number 2, producing modulators or removing modulators. You can do both. That change reaction rates. The concepts include feedback inhibition, some post-translational modification, and cofactors. So we're going to show you some examples, or I may get to these later on. I'm going to do a lot more later on, but certainly these two for sure. Feedback inhibition is just like what it says. It's negative feedback. In this case, what happens is as we go through a series of chemical reactions, each catalyzed by different enzymes, we end up with the final product here, Z, letter Z. Now, Z oftentimes is an inhibitor of enzyme number 1. That way, we don't make too much B, too much C, and certainly not too much Z, and thus waste energy and waste hard to get nutrients. So what this does is once Z meets the sort of metabolic threshold needed, it will come back and it will inhibit enzyme 1. Turns out there are even nested pathways, for instance, like C can come back and feedback. You have nested feedback inhibition loops sometimes. And those are situations where the amount of intermediates and the amount of product are very, very carefully regulated. So final product will often do this, a negative feedback pathway. Sometimes the intermediates will as well. Let me show you a billion-dollar idea. Very, very simple, but worth tens of billions of dollars, if not more, maybe hundreds of billions of dollars before it's all said and done. If we were to look at cholesterol biosynthesis, so we start out here with a precursor, and we're going to make cholesterol, and your body makes its own cholesterol if you don't get any. It's so important as a membrane fluidity buffer and as a precursor for other hormones like testosterone and estrogen, that your body will make cholesterol if needed. If you eat something that has cholesterol in it, well, the cholesterol will serve as an inhibitor on the very, very first step of the cholesterol biosynthesis, and that just happens to be shown here. And you don't have to know this reaction, but in any case, we start out with this HMG-CoA. like acetyl-CoA, but it's an HMG. I forget what the HMG stands for. In any case, there's an enzyme called HMG-CoA reductase. Some biochemists discovered it, and they said, hey, it reduces HMG-CoA and makes a molecule called melanate, or mevalonate, excuse me. In any case, this is the first step, and cholesterol will come in and inhibit that. So here's cholesterol right here, comes in, boom, shuts it down. You might have heard of a condition out in the world called familial hypercholesteremia. Familial hypercholesteremia is where cholesterol in the body does not feedback inhibit the isoform of enzyme that person has. That's why it tends to run in families. You inherit an enzyme that is resistant to cholesterol feedback inhibition. And so in addition to all the cholesterol you're probably eating in our lovely Western diets, your body is also making more cholesterol. And so you have high cholesterol levels in the blood, hypercholesteremia. Familial means that you're inheriting the gene, the enzyme, from someone, one of your parents, or both in some cases. So somebody had the idea, well, maybe we can invent an inhibitor that will mimic cholesterol, or if somebody just has a high cholesterol diet, maybe it'll augment the feedback inhibition. And that's where the statin drugs come from. And each of the drugs is a billion-dollar seller. It's very, very important for staving off heart disease and vascular disease by limiting the amount of cholesterol that appears in the blood. A very, very simple idea. Let's inhibit that first step just like natural cholesterol will, and boom, billions of dollars in pharmaceutical sales. On the other side, we have what are called cofactors, which are usually activators. They are modulators. Some will turn things off and some will turn things on, and maybe the same thing will turn some things off and some things on, depending on the protein. You know, it gets kind of complicated. Let's keep it simple. Cofactors modulate enzymatic activity. They bind to the enzyme non-covalently, and they influence the activity of that given enzyme. And a couple of real common ones also serve as second messengers, if you remember cell signaling. We'll talk about that later. Second message. Second message is often a cofactor activator of some kind or a cofactor modulator. Calcium's very, very common in this fun molecule that some people will call CAMP, but the correct way of pronouncing it is cyclic AMP, cyclic adenosine monophosphate. Have you heard of adenosine triphosphate? This is a metabolite of ATP. It's a monophosphate, and it's been made into a ring shape. Let me show you a picture. So here's adenosine monophosphate. So one phosphate, and here's the adenosine side, the nucleotide base A and the ribose sugar. Here is cyclic AMP. That phosphate has been flipped around and actually binds the molecule, the sugar twice. That's why we call it cyclic. Yes, there's a ring here, there are rings here, but when they say cyclic, they're referring to this ring right here. Very, very common second messenger, very, very common protein modulator. So let's look at calcium and show you what I mean. So let's say we have a protein here. It's a protein, it's got a given shape and so forth. And it's sensitive to calcium, and so calcium comes along and you see the calcium's binding to the protein in a non-covalent fashion. Changes the structure. Look at now, it looks like a fish hook or a letter J, whereas this one looks more like a letter E or something. It has significantly changed the shape of the protein, and now we have these binding sites that are now available. And so the protein can now bind and interact with whatever its substrate is. So that's an example of where calcium is bound, and it's sort of like unrolling the red carpet for the substrates to come in and bind. Okay, so we've done manipulating concentrations, modulators, though we will get back to post-translational modification later on. It's very similar story to these. We're just going to be doing more covalent modifications and so forth then. So now let's move to number 3, using different enzymes to catalyze reversible reactions. Again, hang tight in this next one because this one can be a little tricky. Many reactions have a need for speed. I want you to write that down for this slide. Need for speed. When the same enzyme, you see this enzyme, carbonic anhydrase, again, another biochemist discovered this one. Carbonic anhydrase is standing next to this arrow. That means it is facilitating the going from carbon dioxide and water to carbonic acid, H2CO3. Carbonic anhydrase can also catalyze the other direction. This is one of the fastest catalyzed reactions in the human body because second to second we need to change the ratio of carbonic acid and CO2. Most of your carbon dioxide that you carry in your blood is in the form of actually bicarbonate, which is a dissociated form of carbonic acid. We'll get to that chapter later on. But when you exhale, just exhale right now, 2/3 of that CO2 that you just exhaled just a split second ago was carbonic acid. It wasn't even carbon dioxide. So when you have the need for speed, you have the same enzyme catalyzing both directions. The need for speed. On the other hand, if you are, let's say we're a prehistoric human being, you had to work very, very hard to get your calories. And so it was very, very important that once those calories reach the cell, they don't get back out. You don't have enough calories to begin with. You want those every calorie to count, right? Well, in this case, this reaction is catalyzed by an enzyme in only one direction. Hexokinase cannot catalyze the reverse reaction. Hexokinase is an enzyme that can transfer a phosphate onto glucose. Most kinases are actually protein kinases. This is actually an unusual one. Hexokinase actually catalyzes the transfer of phosphate from ATP onto glucose. If you remember your, learning about glycolysis, remember that phosphorylation of glucose is one of the early steps, and that's why you invest energy into the system. You're phosphorylating it in order to set up for breaking it down into a 3-carbon molecule. But hexokinase can only go this direction. That way, it doesn't catalyze the reverse. If it catalyzed the reverse, if we had a lot of glucose 6-phosphate, we'd start losing it, and that would reduce our efficiency. So using only an enzyme that can convert one direction is an important control step. Turns out, though, that there are a couple of cells in the body that release glucose. Your liver cells release glucose, and some of your kidney cells release glucose. In that case, they happen to have the other enzyme. It's called glucose 6-phosphatase, not phosphorylase. Don't get that mixed up. Phosphatase. The glucose 6-phosphatase pops that phosphate off. So hexokinase puts the phosphate on and it uses ATP, so the ATP should be the reactant here. But you're transferring that phosphate and now you have a phosphorylated glucose. But in those cells that are going to release glucose back out into the bloodstream, your liver cells, your kidney cells, they will turn on glucose 6-phosphatase and it'll pop off that phosphate off the 6th position back into glucose so that you can secrete glucose. So now you have a form of control. You create an enzyme that can only catalyze one direction. So most cells are like this. 99.9% of your cells in your body are like this, but the liver and the kidney cells that are responsible for releasing glucose when you're low glucose have both enzymes actually. So a kinase, and this is an important definition for you for moving forward in this class and certainly down the line, especially if you're involved in pharmacology and that sort of a thing. We take ATP and we have a substrate here. In this case, the substrate is glucose, but in most cases, a substrate will be a protein that we're going to turn on or turn off by doing this phosphorylation. And so the kinase will facilitate the transfer of that phosphate. So we go from a triphosphate to a diphosphate, and that phosphate's been tacked on at a very, very specific site on that protein. Kind of like a doorbell button for a door. You know, you have to put that phosphate in just the right spot for the right events to occur. So they're very, very specific. On the other hand, the phosphatase will start with that phosphorylated substrate, and it doesn't need ATP. We're not going to phosphorylate ATP most of the time. You're just going to pop that phosphate off. And so that's a phosphatase and a kinase. We are taking we're cleaving that bond off of the phosphate. Here we're transferring that bond to the substrate. Kinase, phosphatase. Now let's go to step 4, compartmentalizing. Let's look at an example. I'm going to use a model system here. Our model system for compartmentation for regulation is making ATP. So ATP is very, very important for transferring energy and storing energy in a given cell. Has that high energy phosphate bond. So we have aerobic, which is a 3-phase multi-step thing that you guys learned about in 165. We also have anaerobic metabolism. It's also multi-step in nature, and again occurs in a given area. These 3 major areas all are occurring in different places within the cell or within an organelle. So there's that compartmentation. And so here's our picture here. Glycolysis occurs in the cytosol. Citric acid cycle occurs way down deep inside the mitochondria. And the electron transport uses the inner mitochondrial space where citric acid is, but also uses the intermitochondrial space between the 2 membranes for that proton gradient that drives ATP synthesis. So we see compartmentation and 3-dimensional regulation at virtually every level of this process. You already knew it, you just didn't realize it. And here's just a fine picture. Here's citric acid cycle. This is way down deep inside the mitochondria. So we're doing this and we're creating this proton gradient, and we have to have that gradient. If we don't, there are actually cells in our body that undergo citric acid cycle but don't do much electron transport. They're thermogenic tissues. We talked about that. Brown fat, remember in the previous lecture? We can also do things like, for instance, we can inhibit this transfer. If we uncouple citric acid cycle with electron transport, that's how cyanide does its thing. There was also a molecule that was used as a weight loss supplement for a period of time called 2,4-dinitrophenol. And people started dying because they were taking too much of it. Why? Because they're like, oh, if I lose a little bit of weight with some of this stuff, I could use a lot of weight if I use a lot of it. Well, they would die of basically the equivalent of cyanide poisoning because they were uncoupling and their body couldn't make ATP. Compartmentation, though, that's the key thing here, compartmentation. 3-dimensional regulation of proteins that catalyze these reactions. And since I've already sort of alluded to with the cyclic AMP, the ratio of ATP and ADP, I might come back to that at some other point, but right now I don't have any plans to talk about that. Let's now talk about post-translational modification and some of this, because this is stuff that's going to be important to you, especially if you're involved in healthcare and pharmacology. Post-translational modification is the changing of the shape, changing of the functional groups that are added or subtracted to a protein after it's been translated. That is to say, translating mRNA into a peptide sequence. So we have folding and cleavage. We may add or subtract molecules, and we may covalently modify them. So that's way down here where folding is involved, maybe sometimes cleavage, addition of functional groups, sometimes lipids or sugars, phosphates, other functional groups, and or the presence or absence of cofactors and polymers, other peptides. So here's your list here, folding. This is like folding, but it stabilizes a fold. It's a cross-linkage. It's a covalent bond between 2 sulfhydryl groups. We call this disulfide bond, and it stabilizes a given fold, a 3-dimensional structure. The more of these, the more robust the folding structure is. Sometimes proteins need to be very robust this way, others not so much. Cleavage, chopping it into smaller pieces. addition or subtraction of functional groups, and then polymeric situations where you have other peptides and you create a multi-protein complex. So here's folding control, and folding control is associated with a lot of diseases, not the least of which diabetes, maybe Alzheimer's, other types of misfolding situations. So we normally, when we translate, we create a peptide, and you have chaperone proteins and things that fold it correctly, and it gets processed out into in the ER and maybe in the Golgi, and then sent to wherever it's going to go. Occasionally, though, these folding proteins don't do it right, and they misfold. And when they're misfolded, the cell can actually recognize that. It has a system designed to identify misfolded proteins and destroy them so that you don't have misfolded proteins. Turns out many diseases, and diabetes is one of them, where, like, for instance, when you're making insulin, you misfold it, and for whatever reason, you can't get rid of it. And so the misfolded protein builds up and starts injuring the cell, creating inflammation and/or cell death. A wonderful example is the mad cow disease, Jakob-Creutzfeldt syndrome in humans. There's a protein called a prion that's actually the disease agent. It's really wild. If the prion gets misfolded, it will actually promote additional misfolding of proteins. And this is occurring in neurons or in glial cells, and that's where you get that mad cow syndrome, because now the neurons are not working properly, because now you have this buildup of misfolded proteins in the neurons. So this diseased prion actually facilitates additional prion creation, thus reinforcing. It's like a positive feedback cycle for a disease in this case. And what's really interesting, if not a little bit disgusting, there was an article a few years back about how cannibal tribes in places like South America, in the Amazon basin, or in certain areas of Africa, and even in Asia, cannibalism is not unique to any one ethnic group. It has occurred over time and in various ways. It turns out that in human populations that have engaged in long-term cannibalism, they seem to have a protection from this disease. People tend to get Jakob-Creutzfeldt syndrome from eating neural tissue from usually cattle. That's why we destroy a lot of cattle when there's a breakout of mad cow disease. You'd want to get rid of any cattle that have the disease agent because it can cause it in humans. Well, it turns out that contaminated meat, the neural tissue that shows up in the diet of people who've been in tribes that have engaged in cannibalism, are more resistant to Jakob. They have much less. They have evolved a protection against that prion disease. Pretty amazing stuff. And folding requires appropriate bonds and cross-linkage. Okay, you have to have the right bonds in the right places. And these bonds are dynamic. They can change based on the ionic strength of where the protein is located, or the pH and that sort of a thing. But the four major bonds, we certainly have the covalent bond, which is in the primary structure. But we also have covalent bonds in the form of disulfide bridges, S covalent bond S. Very, very strong. Folks who have extremely curly hair, if they want to straighten their hair, often have to use a lot of heat or strong chemicals. And that's because what you're doing is you're breaking the disulfide bridges. Or if somebody's getting a perm, we don't see that a whole lot anymore. People don't get perms all that much anymore, but some people still do. When you do a perm, you're doing the opposite. You're creating the conditions in order to create disulfide bridges in the keratin proteins in the hair. Hydrophobic interactions, hydrogen bonds, all of these bonds have to be have to be in place correctly, and that's why pH and ionic strength and things are so important. But there's that disulfide bridge that we talked about. It's just another example of proper folding for appropriate structure and appropriate function. Protein cleavage is also a very important regulatory pathway. What protein cleavage is doing is that if a given signal occurs, what will happen is you will cleave a protein at a given site. It's usually very, very precise. Like phosphorylation, you have very, very distinct locations where a protease, another enzyme, will come in and snip this peptide. This can serve a very important signaling role and regulatory role. Like for instance, in apoptosis, we talked about apoptosis. One of the enzymes that's involved in apoptosis is caspase-3. Caspase-3 is normally kept in an inactive state unless there's a signal of some kind. And when the signal occurs, we cleave this peptide into 3 different units, and now we have, and those units will reassociate in some cases, and now we have an active caspase-3, which is also itself a protease, so it's going to start cleaving other targets. So we went from inactive to active. Where you see lots of protein cleavage is in blood clotting. The series of steps going through blood clotting, and in fact many hemophilia patients, they have a problem with this cleavage cascade getting to a clotting paradigm. We also see this in protein hormone regulation. For instance, insulin. So we'll talk about insulin at a future point at some time. Again, we cleave this in order to turn it on. In other cases, we might cleave proteins in order to turn them off. You can also covalently modify a protein by adding, or in some cases subtracting, functional groups and other molecules to them. I say adding and subtracting because sometimes you'll have a modified protein and it's inactive, and you'll take the functional group off and it'll become active. This is modulation. It can be up or down. So phosphorylation, we talked about that before with our kinases. Kinases will phosphorylate a substrate using ATP. Usually unidirectional. Glycosylation, an example of here where we add carbohydrate chains, polymers to these proteins. This is exactly what we do with our A and B antigens in the blood. We will add these carbohydrates when the protein is in the Golgi, and we'll fold those, we'll flip those out on the outer surface of the cell as identification for self and non-self, et cetera, et cetera. Or we have lipids, and I can see some lipids kind of sticking down in here. You can kind of see some lipids here. Here's some carbohydrates, by the way. This isn't the best picture in the world, but lipids may help us, for instance, localize a protein to one side or the other of a membrane. It could be the plasma membrane or another one, and so forth and so forth. The story remains the same. It just depends on the molecule you're talking about. Lastly, this idea of assembly into polymeric proteins, where if you have these 2 subunits separated, the enzymatic function doesn't occur. But if they associate, and usually it's a non-covalent association using hydrogen bonds and ionic bonds and van der Waals, hydrophobic interactions. Once those units associate, then we have active enzymatic complex. So we can regulate the activity of the enzymatic activity by whether we have or how much we have of both subunits. And that's it for today. So here's our very, very brief summary since we're at 33 minutes. Make sure that you see what we talked about in each of these areas. And again, this is pretty brief. Hopefully, with the aid of the PowerPoint and or this lecture or what we talk about in class, you'll be in great shape understanding what is Introduction to Biochemistry. It's a bunch of simple ideas all kind of working together at once, and that's what makes it tricky. Hopefully you'll agree with me. I know that that's not always the case, but hopefully you will. I really want to make this more accessible to you because the more you can understand this stuff, the easier it'll be to have conversations with things like nutritionists or medical doctors or pharmacologists or your pharmacist if you're working with one, or your patients even for that matter. Why would you take a certain drug? Give them a little background, help them they'll be more compliant about their care. All right, thanks for your time, and I will see you in Chapter 5.